Hello folks, I want to do a short video for you on, now that we've talked about what fuels or what molecules the muscles use to fuel activity or to allow contraction to occur. I want to talk about how we vary which type of fuel we're using or which type of molecules we're using based on the intensity of the activity. And what we mean by intensity is the ATP demand, like how much ATP is being demanded by a working muscle, by a contracting muscle. And if the absolute demands over some interval of time is very high, we call that high intensity. And if the absolute demand is low, we call that low intensity, and moderate or medium intensity for sort of an intermediate amount of ATP required. So we're going to think about that. And recall, we've talked about maybe 4 different ones, but try to think about definitely the 3 places where ATP is used during muscle contraction, right? Now, we've talked for a little bit now about the sodium-potassium pump, right? And that's important for both regulating the charge distribution across the membrane and maintaining the transmembrane potential for all cells, not just muscle cells. But then it's also really critical for reestablishing that potential difference, that resting potential after a single action potential sweeps across a muscle fiber, right? So once we get down to that hyperpolarization phase, getting us back up to that resting potential, that's all sodium potassium pump, and that's all because it's a pump. It's driven by ATP, right? So you could imagine that when we're doing something like summation, right, where we're getting lots of action potentials per unit time, that's going to be a higher demand on those sodium potassium pumps. Where else have we seen it? Where else have we seen a use of, or a place where we use ATP during muscle contraction? Hopefully you're able to say the calcium pumps on the sarcoplasmic reticulum that are responsible for grabbing calcium from the sarcoplasm and shoving them back into the sarcoplasmic reticulum or to the terminal cisternae of the sarcoplasmic reticulum. Right? That's a major use of ATP. And you can imagine, right, as you get more twitches per unit time, that's more times that those sodium or that those calcium pumps have to work to reestablish that, or to remove calcium from the sarcoplasm and put it back into the terminal cisternae. The other major place during a twitch contraction has to do with the myosin and actin. Hopefully you said detaching the myosin head from the actin filament, right? Remember that myosin head remains attached to an actin filament. Even after the power stroke, it remains attached until a new ATP comes and liberates it and allows it to go through its recovery stroke and reattach. Right? So that's another use of ATP. So within a single twitch contraction, there are all these uses of ATP, and I know I'm forgetting one too. I just named kind of the top 3, but there's another one I'm not thinking of. Hopefully you can think of the other major use of ATP. Oh yes, right. Let's just pop in here real quick, and we'll remember that another major use of ATP is in glycolysis. So in order to complete glycolysis, the anaerobic, so no oxygen required, anaerobic metabolism of glucose, in order to yield a couple of ATPs, right? Doing this process, going from glucose to these 2 pyruvate molecules down here, that requires a little bit of ATP. Of course, we net 2 ATP out of it, but it requires a little ATP to start. Okay, so that's another use of ATP. All right, so let's now just check out what we're going to talk about in this recording. Now that we've summarized all that from last time and thought about ATP use, and now we're thinking about the intensity of ATP use, the learning outcomes we're really going to be focusing on, and I'm just going to pop over to that page, are what type of activity, during what type of activity or what intensity level of activity are we primarily using carbohydrate or fats, right? So let's go down here. So we've already talked about 13, right? Now we're going to be doing 14 in this video primarily. And then the last video, we're going to do 15 through 18 here. So we're going to be thinking about what fuel is used and when, during what level or intensity level of activity, and what are the byproducts of both aerobic and anaerobic metabolism, and how do those affect the muscle's ability to continue working? And so how does it affect or impact muscle fatigue, right? Okay, now jumping back over here, we've got our fuel use varying with intensity of activity. So let's talk about those different intensities. And we mentioned this in the last video. So I'm just going to show you kind of what's going on within a muscle cell when we've got this different intensity of activities. So we're going to start out here with low intensity, right? Or more or less a resting muscle, or you could even think low intensity activity. What's happening in a muscle cell? Okay, well, if it's at rest or just metabolizing very slowly, just kind of maintaining itself, it's importing all the things that the vascular system is bringing to it, all the things of the blood vessels are bringing to it, right? So fatty acids probably from food that you've eaten, and glucose from food that you've eaten. If you haven't eaten in a while, you're still getting this fatty acids and glucose from triglycerides that are putting fatty acids into the blood so that they can be taken to muscles, and glucose, which is coming from the liver and being delivered to skeletal muscle. We're getting the substrates, right, to make ATP, and the other substrate, of course, is the oxygen gas, right? This is required for aerobic respiration, and so the efficient creation of ATP. Okay, so all that's being imported. Notice these arrows. Glucose is being imported, and it's being stored as this very large polymer of glucose called glycogen. You remember from Biology 160 that glycogen is the preferred storage form of energy, or one of the preferred storage forms of energy, and especially of glucose, for animals and for our closest relatives, the fungi, right? The mushrooms that are all coming up right now outside, they store energy as glycogen. So that's happening. We're also using those fatty acids and oxygen in order to fuel the creation of ATP, right? So those fatty acids are being imported into the mitochondria. Remember, there's nothing we could do with them in the sarcoplasm. And as long as we have oxygen, that final electron receptor, we're basically carrying out the Krebs cycle or the citric acid cycle in the mitochondria, and producing lots of ATP, which we're banking. We're also producing lots of creatine phosphate, which we are banking, right? So we're basically creating fuel storage for these components that fuel muscle contraction: ATP, creatine phosphate, and glycogen. Now, what is a byproduct here? A byproduct is carbon dioxide. You know, that is a byproduct, as well as water, metabolic water. That's a byproduct of all aerobic respiration, is carbon dioxide. Right? So in this sort of a state, muscles are primarily using fatty acid to fuel all of their activity, and they're also using that fatty acid to hopefully bank some ATP and creatine phosphate. So we're making reserves. Now, what happens when we have moderate muscle activity? We still have all that same stuff going on. We have glucose being imported to, we have fatty acids being imported and oxygen being imported. All of that is still happening. There's still plenty of oxygen around at sort of moderate muscle activity. But now, after if we're doing this for a while, we exhaust our ATP sources, we exhaust our creatine phosphate sources. And so now, remember, we have to go somewhere else to make our ATP. So we're still doing this. We're still importing fats and still metabolizing those aerobically to make ATP. But we're also breaking down our glycogen into glucose, carrying out glycolysis, where we get these two ATPs, right? And then carrying the pyruvic acid or pyruvates, converting them to acetyl-CoA and carrying those into the mitochondria to also go through the electron transport chain and the Krebs cycle to make a lot of ATP, right? Relatively a lot of ATP out of this one glucose molecule, right? So we're still doing a lot of aerobic metabolism. But now rather than banking a lot of this material, we're breaking it down, right? And we're making now more carbon dioxide because we're doing more activity. So now there's more of this byproduct of carbon dioxide, which does tend to acidify muscle tissue. And what else are we doing? Well, if we think about moderate muscle activity, we are using more ATP, and we're doing more glycolysis. We are doing glycolysis now, and this process of glycolysis. Remember, we saw last time that we have to have those electron carrying molecules grabbing the hydrogen nuclei and carrying them off from here to the mitochondria to enter the electron transport chain. That process of stripping, right, of phosphorylating ATP liberates a hydrogen nucleus and an electron. So when those are when that ATP is pulled apart and that glucose is pulled apart, in the sort of the net of that is that we get some free hydrogen nuclei. We get some free hydrogen ions. And if you remember from Biology 160, as you increase the hydrogen ion concentration of a solution, what happens to its acidity? What happens to its pH, right? Acidity goes up, pH goes down, right? So as we do more of this glycolysis, we're actually producing more hydrogen ions, which acidify the blood or the tissues and blood, and we're producing more CO2, which do the same. They contribute to acidification of the tissues. Okay, so sorry. So now let's go to peak muscle activity. This looks weird now. Where did all this importation of stuff go? It's still happening, right? We're just showing what's primarily happening, right? So we're still, at peak muscle activity, your heart's beating hard, you're breathing rapidly, you're still importing as much oxygen as you can, right? You're still importing fatty acids, though you're probably not using very many of them, and you're still importing glucose, but primarily what you're doing is this lots of glycogen breakdown into glucose, because now we're at peak muscle activity. That means muscles are working very, very hard. You're stressing them, right? And what does that mean to be stressing them? You're stressing their substrate availability, right? They have a certain amount of substrates that they store within the muscle and that they're able to deliver during muscle activity, during contraction. And now you're stressing that system, right? So you're using lots of creatine, phosphate, whatever's there to make more ATP. You're doing this glycolysis to make ATP, and your ATP demand is so high, right, that a large fraction of this glycolysis that's happening doesn't result in those hydrogen nuclei being carried by those electron transport molecules, or those helper molecules, not electron transport, but electron shuttling molecules. It doesn't result in those electrons being carried off to the mitochondria, which we would see down here, right? And it's still working, of course, but a good fraction of this is not being carried there for aerobic metabolism. And the reason is because our ATP demand is so high and the electron transport chain takes a while, and we're basically maxing it out. We're delivering as much oxygen as we can to continue fueling that electron transport chain, but we're maxing that out, and we still need more ATP, right? So we're not generating enough ATP via electron transport. We have to make it in another way, and that's glycolysis, and that's the way we're making most of our energy, because our energy demands are so high now. Our ATP demands are so high. Most of it's actually coming from glycolysis. Maybe about peak muscle activity, 66% is being made by glycolysis, just because our mitochondrial ATP production cannot meet our energy demands right now. And so what's happening is a lot of this pyruvic acid is because these electron shuttling molecules are just dropping those hydrogen nuclei back off. they're reattaching to pyruvic acid in a slightly different conformation, making it lactic acid. And lactic acid is not a useful substrate for making ATP by our muscle cells. So now this is just a byproduct. We've got to get rid of it, right? So we have to export this lactic acid. We got to get it out of our muscle tissue, take it back to the liver. Remember this, we never named it before, I think, but it's called the Cori cycle. But it's where we have to take that lactic acid back to the bloodstream, ship it to the liver. The liver turns it back into glucose, ships it back to the muscle cell. Takes a long time and lots of energy. So we're getting a lot of ATP production via glycolysis and a lot of production of this lactic acid non-useful byproduct. So we got to get rid of it. And of course, we're also, because we're maxing out our mitochondrial activity and our ATP production via aerobic metabolism, because we're maxing that out, we're also producing a lot of carbon dioxide, right? And of course, we're producing a lot more free hydrogen ions during this process that we talked about earlier. So this glycolysis, remember, releases free hydrogen ions into the cytosol. So all this increased ATP consumption, primarily via this glycolysis, increases the hydrogen ion production, right? So that is what acidifies our tissue, the carbon dioxide production and this free hydrogen ion production. Primarily, that's what acidifies our muscles and our blood. I just want to pause here because this is something that many students come into the class believing that lactic acid acidifies muscle tissue. No, it doesn't. It does not. And there are a number of well-done scientific studies now that demonstrate that lactic acid does not acidify muscle tissue. And in fact, though we're not going to get into it, it acts as a buffer to prevent acidification of tissue. So it's not lactic acid that increases acidity of muscle tissue. It's free hydrogen ions that come primarily from this glycolysis and this transferring of phosphate from creatine phosphate to ATP. And that's the primary acidification of muscle tissue. You'll even see it in your textbook saying something about lactic acid and acidity. And in muscle tissue, that is not what happens. So you've heard it before. It may even still say it in your textbook. It does not. It's not physiologically what's happening. And the last thing I'll say about that is the worst. Maybe it's not a bad paper, but the worst thing that was ever published about this was a simple graph showing increase in lactic acid production coincident with decrease in pH. And there's more or less a linear association there, a linear correlation. So that as lactic acid, and I think lactic acid was on the x-axis and pH was on the y, right? There was this increase here in lactic acid, and then this was showing pH, increasing pH. So of course, we're starting here at normal pH and going to low pH, right? That would be acidifying. There was this beautiful negative correlation, right? And that's all it was. It's a correlation. Two things that happen to co-occur. It's not causation. So saying that lactic acid, the inference they made was that lactic acid increases in lactic acid concentration caused decreases in pH. And that's bogus. All they did was measure those two things. They actually didn't measure anything about how that lactic acid would be decreasing pH or increasing acidity. And that's really important, this difference between correlation and causation, because people do it all the time. Look at two factors they choose to measure, and if they find an association, they say, oh, look, it's causation. No, it's not. And it's not in this case. And this is really critical that you understand that sort of general principle, but more specifically in this case, that increases in lactic acid don't cause increases in acidity. Okay. It turns out that it's this drastic increase in free hydrogen ion concentration. Okay, I'm off my soapbox. Now, let's look at how this all affects muscle fatigue, right? So you know that you may be working really hard and you feel your muscles start to burn, right? Or maybe you just can't lift or do the activity you've been doing. Why does that happen? Why do muscles fatigue? There are a lot of causes. In the case of, it turns out, endurance activity, where you're doing some activity that's easy for your muscles to do, but you're doing it for a really extended period of time. It can be due to the disruption of ion concentration gradients across the plasma membrane, and especially across the T-tubule, the membrane of the T-tubule, right? Remember, those T-tubules are not very big, and so things diffusing into and out of T-tubules, it doesn't take place as easily as stuff sort of diffusing around the outside of a cell, right? Because they're interacting with all those transmembrane proteins which are charged, and so it takes a while for diffusion in and out of T-tubule to take place. So it's really, studies have shown that we get pretty massive disruptions in especially potassium concentration across T-tubule, as well as calcium concentration, right? And the other thing you can get is over repeated stimulation of a muscle fiber. You can get calcium disruptions across the terminal cisternae, so across between the terminal cisternae and the cytosol, but only after pretty long periods of exercise. It's possible that you could run out of the energy yielding substrates, and this can happen in very long distance races where you start burning proteins. Okay, so you could run out of these energy yielding substrates, but typically this comes at the end of exercise, when we're actually dipping into burning proteins, which we can metabolize proteins in order, some proteins or components of proteins, to make these energy yielding substrates. So that can happen, but really it doesn't contribute greatly to muscle fatigue. Insufficient oxygen, that can definitely happen sort of at any time, contribute to muscle fatigue, but that's always coupled with metabolic acidosis, right? Metabolic acidosis means the reduction in pH, the increase in acidity that muscle tissue experiences, caused by that that increase in hydrogen ion and CO2 concentration, right? So metabolic acidosis is we think the primary cause of muscle fatigue. And it, for a variety of reasons, like, okay, well, what is this acidity, what does that disrupt? Well, you know all about what problems acidity can pose for proteins. Remember from Biology 160 that you saw that proteins have an acidity at which they're most active. And as you get away from that acidity, either more basic or more acidic, away from that ideal pH, proteins become less active, right? And for a variety of reasons. You also know that, if you increase the ion concentration, whether positive or negative, of a solution, you're going to definitely disrupt the binding of salts together, the binding of even proteins, the ionic bonds of proteins, right, as well as the hydrogen bonds of proteins can be disrupted. So acidosis, actually, we know that it influences calcium binding efficiency. So that means that calcium is binding to troponin not as well, right? If it's not binding to troponin as well, we're not getting exposure of the active sites, right? And so we can't get myosin grabbing onto actin. And so this is one major cause of muscle fatigue. We also know that it reduces rates of ATP hydrolysis. So it simply reduces the rates of all of the ATPases out there. So the myosin, right? It reduces rates of myosin splitting ATP and detaching from the actin head. It reduces rates of sodium, potassium pump, and calcium pump. So all ATPases stop working as efficiently in response to this decrease in pH or increase in acidity. Okay, and this is just to show you some data that supports that. This is looking at the concentration of ATP on the x-axis. This is the velocity of muscle shortening, so how quickly a muscle contracts, right? a given muscle fiber. So this is obviously very small, micrometers per second, but this is a velocity, right? Now you see that each one of these lines is a particular pH. This is about neutral, 7.4, and this is a bit acidic, 6.4, though not terribly, but it is acidic, right? So think of this as like a resting pH value, and this is like exercising pH value. And what I hope you can notice from this, right, is that at the same ATP concentration, there is a enormous difference in shortening velocity across all ATP concentrations, right? When the pH of the muscle tissue is acidic, right? So the rate of muscle contraction, right, the speed of muscle contraction is depressed by this pH, right? By this exercising pH. And for those reasons we just discussed, the calcium binding efficiency and specifically the detachment rate. And this is actually looking at just detachment rate, right? And this is not looking specifically at pH, right? Here, sorry, this is not looking at pH here. This is just a normal pH. But it's showing you one of those other reasons we talked about for muscle fatigue, not having enough substrate, right, around. And so you can see this is detachment rate, detachment of myosin from actin. And this is concentration of ATP. This is more or less the same as this. This is just in molar and this is in micromolar, but it's more or less the same axis. So you can see here at low ATP, there's a severe depression in detachment rate, slowing the rate at which a muscle can contract. And it isn't until we do plateau up here. Once we get to about here, we're at normal detachment rate or maximum detachment rate that the muscle is capable of. But for a lot of these concentrations of ATP, which we could experience during intense exercise, right, and could contribute to muscle fatigue, especially during intense exercise, is this reduction in substrate. Okay. This is just another look at speed, but also power. So this is looking at power here, the ability to move across some length, right, or across some distance, right, is a measure of power. And what I hope you can see here is that at normal pH, right, and all that you really need to take away from this, at normal pH, there's this power that's related to the force that's generated, right? And at an acidic pH, right, that curve is still there, but it's again severely depressed, right? So a force that would normally produce this power up here, right, this force produces almost no power to move the muscle, right? In other words, if we're thinking about a muscle contracting or enough muscle fibers contracting to overcome a resisting force, it's severely depressed by pH, right? So not just the velocity of muscle contraction which is largely driven by the detachment rate of myosin from actin, but also the force that it's able to produce, right? The power it's able to produce for a given amount of force is severely reduced as well. Okay. All right. Let me know if you have any questions about that. If you need to go back and look at those graphs or if you need some help understanding those graphs, I'm happy to talk you through that. And then in the next lecture, we'll finish this off. We'll talk about the different types of muscle fibers that we have, fiber types that we have within whole muscles, and how those differ anatomically, and what are the physiological consequences of those. And we'll also talk about muscle adaptations to repetitive bouts of exercise. Okay, I'll see you again soon.